Welcome to the TOEFL Listening section. In this section, you will listen to an academic lecture and then answer questions about what you heard. You may take notes while you listen. You will have time to review your notes before answering the questions. Listen to part of a lecture in a university class. You will hear the lecture only once, so listen carefully. After the lecture, you will answer some questions about what you heard. Some questions ask about the main ideas and supporting details. Other questions ask you to make inferences or connections between ideas. Answer each question based on what is stated or implied in the lecture.
Good morning, everyone. Today we will examine host-parasite coevolution and how this evolutionary arms race shapes disease dynamics and biodiversity. By host-parasite coevolution, I mean the reciprocal evolutionary change between hosts and their parasites caused by the selective pressure each imposes on the other. A useful way to frame this is the concept of an arms race and the Red Queen idea. In an arms race, hosts may evolve improved resistance, while parasites evolve greater infectivity or virulence in a directional manner. The Red Queen metaphor describes a different dynamic. Their selection is frequency dependent and often results in oscillatory change where hosts must constantly evolve simply to maintain their relative fitness against parasites. To make these ideas concrete, I will use 3 case studies from different systems. First, the Myxoma virus that was introduced to control European rabbits in Australia illustrates the arms race. The virus was initially extremely lethal, but within a few years both the virus and rabbit populations changed. Virulence of the virus declined in some lineages, and rabbits evolved increased resistance. The outcome was not a stable victory for either side, but a change in both parasite traits and host genetics. Second, the relationship between Plasmodium falciparum and human populations shows balancing selection in hosts. The sickle cell mutation is an example where heterozygote advantage reduces severe malaria risk, while homozygotes suffer disease. This balancing selection maintains genetic diversity in human populations in regions with intense malaria transmission. Third, in plant pathology, the gene-for-gene model describes how specific resistance genes in plants correspond to virulence genes in rust fungi. When a plant population carries a resistance allele, parasites with matching virulence alleles are at a disadvantage, but parasite populations often evolve new virulence alleles, and the cycle continues. At the molecular level, parasites use strategies like antigenic variation, secretion of immunomodulatory molecules, and rapid generation of genetic diversity to escape host defenses. Hosts counter with innate immune recognition, adaptive responses, and high diversity at loci such as MHC. An important idea is trade-offs. Increased virulence may increase short-term transmission, but can reduce host survival and therefore long-term transmission opportunities. Similarly, resistance in hosts often carries a fitness cost in the absence of the parasite. Experimental evolution studies with bacteria and bacteriophage show both escalation and fluctuating selection. In some experiments, phage evolve broader host range while bacteria evolve costly resistance mutations, illustrating an arms race. In other experiments, allele frequencies cycle, matching Red Queen dynamics. The Geographic Mosaic Theory helps explain why coevolutionary dynamics vary across landscapes. Some populations are hotspots of intense reciprocal selection, while other populations are coldspots with little coevolutionary change. Local adaptation emerges when parasites are better at infecting local hosts than foreign hosts. These principles have practical implications. In agriculture, understanding gene-for-gene interactions guides deployment of resistance genes to slow pathogen adaptation. In public health, recognizing antigenic variation informs vaccine design, and in conservation, acknowledging coevolutionary processes can shape strategies for managing emerging diseases. We must also consider long-term consequences. Coevolution can maintain high levels of genetic diversity in host populations and drive speciation in parasites. It can also produce surprising outcomes, such as reduced virulence, if transmission depends on host mobility or density. Finally, I want to emphasize a conceptual takeaway. This is why the Red Queen metaphor is useful. Hosts must constantly evolve just to maintain their current fitness relative to parasites. In summary, host-parasite coevolution is a dynamic, context-dependent process shaped by genetic architecture, ecological factors, and life history trade-offs. It is central to understanding disease emergence, control, and the distribution of genetic variation in nature.
Listen again to part of a lecture, then answer the question.
This is why the Red Queen metaphor is useful. Hosts must constantly evolve just to maintain their current fitness relative to parasites.
This concludes the listening section. For more TOEFL practice tests and study materials, visit Spinta.org.